Assalamu alaikum guys. Today I am going to tell you how to extract the tissue specific expression data of the model plant, for example Arabidopsis thaliana. For this purpose today I will use a database which is known as a TraVA database. So just type the TraVA on your Google tabs, then this interface will appear. Just click on the TraVA, then it will lead to this page. Here you can see the model plant, the Arabidopsis thaliana. When you will click on this, then there is the four other plants about which you can get the data from here. So this is I'm interested in the Arabidopsis thaliana, so I will select this one only. So here is the tab like how to use this, and the samples, browse DB and contact. So here you can see that this database of gene expression profile in plants based on the RNA-seq analysis. So here the data in this website is the data which has been extracted from the RNA-seq analysis. So here when you will click on the how to use this, then there is a whole procedure which you can follow step by step. So I'm not going to tell you each and every detail. If you are interested, then you can search this, but I will directly go to the browse database, like how we can search our data. I will click on this. So here is the interface. For example, this is a meristums, a flower and flower part, and here is the for example shoot apical meristem tissues. So here it will show the some color from which we can detect like the expression of the gene. So what we have to do is just we will enter our gene ID there. So I will copy my gene ID. So as you know that we have already extracted our gene IDs from the their database or from the some other websites such as the NCBI. So I will copy my gene ID and I will go to my database and I will just insert it there. So here you can see that there is the read count normalization type. So it is met, it is fine. Here is the expression, like it will give you the expression ranging from 0 to the 1. 0 shows the down regulation and the 1 shows the up regulation. And here is the software, but you don't need to go into the detail of this if you are just going to extract the simple data. So I will just browse this, and here is the some data related to this. So it is showing the some color. We can download as the Excel file of this. So now this is only for the one gene. So for example, here the expression is 0.01, somewhat upregulation. For example, very minute upregulation. For example, 0 shows the downregulation and it is 0.01. So if you are interested in searching a lot of gene IDs, so I will recommend you that just search the 10 to 15 genes at a time. For this purpose, I have get my gene IDs and I will copy all of them. Here, the one thing is most important, that the gene IDs should be separated by a space. So, and there should be no sign of greater than as we use in the FASTA format. So I will copy them and simply I will go to the Google and paste my gene IDs there, and I will browse it. So it will tell me the detail of my gene. Here you can see the expression For example, this color shows that it is highly upregulated, and white shows that this is highly, they show the no expression. So here is the down regulation or highly upregulation is there. So you can easily get the data like that. So here is the tissue, for example, petal of the mature flower. So the RAP 2.4 gene shows some upregulation in petal of mature flower, while if we look into the RAP 2.9 gene, so this gene showed do not show any expression here. So you can just simply use the snipping tool. So for to show you, I am just using this, so for the snipping tool, and you will just cut this and then paste it, and then you can use this for your uh, research. So this is how we can get the data. And the other thing is that if you are interested in just getting the data with not interested in doing in such type of picture, so you will just click on the download as XLS. You will just click this and it start downloading the file. This will be the Excel file. When I will click on this, then we can see the expression of all the expression of the genes in the tissues. So I will go back there, and if I will click on the show read counts, it means that if I am interested in getting the gene expression, how much the gene expression is. So I will click on yes. So here is a gene expression. For example, 0.81 shows the highly upregulation and 0.00 shows the downregulation. So in this way you can get the data from the TraVA database. The TraVA stands for the transcriptome variation analysis. And from this database you can also get the data about the tomato. So, and the other crops, about the other crops, there is no data about them there. Thanks.